All right, good morning. My name is Mary Leonard. I'm chair of the Department of Pediatrics, and it's my pleasure to welcome you to Pediatric Grand Rounds. We'll have a few announcements. First is there's the text code on the left for CME credit, and we'll put that in the chat for your convenience. Next slide. We're so excited to have Shakita Bell come and give us Grand Rounds next week in honor of Native American Heritage Month. Shakita has been an extraordinary advocate on the national stage for Native American youth. And I thank the Grand Rounds Committee for this wonderful invitation. Next slide. And then the following week, we're so proud to have John Bernstein present his research with the Undiagnosed Disease Network. This national initiative has really transformed the way we take care of genetic patients, identify disorders, and come up with effective therapies. Next slide. And I hope everyone is well aware of this workshop of the Clinical Research Support Office. The folks on the left here have moved heaven and earth to overcome some of the red tape that can hamper clinical research. So this is a really important workshop for all of our faculty, but importantly for clinical research coordinators and staff. Will be time well spent. Next slide. And we hope you all know about our Drive in Research Pipeline. This is a program that we launched over the summer, providing the undergraduate with a research opportunity for our minority, first generation, and low income students. This was really a remarkable accomplishment that this was accomplished over COVID, and Dean Miner has recognized this as one of the many ways in which pediatrics is advancing our mission around this area. And so we're seeking faculty mentors for 2022. There's a stipend that funds the undergraduates to work with your research programs. Next slide. And it's that time again to think about the Pediatrics Research Retreat on April 29th. Please mark your calendar. The poster, the oral presentation and poster abstract submissions are due January 16th. We really encourage our trainees, in particular postdocs and junior fellows and faculty, to contribute abstracts. Next slide. All right, so moving on to this morning's Pediatric Grand Rounds. I am so pleased to welcome Lance Prince. Lance just celebrated his 1-year anniversary here in the department, and this is a long-awaited Grand Rounds by Lance. We know this is something that's going to be of interest across the School of Medicine, given our commitment to precision medicine. So Lance is professor of pediatrics, chief of the division of neonatal and developmental medicine, and co-director of the Johnson Center for Pregnancy and Newborn Services here at Packard. I could spend a very long time on this introduction and probably embarrass Lance, but I just want to make a few points. One is that he is widely recognized as a world-class physician scientist, dedicated clinician, generous mentor, and visionary leader. We were so fortunate to recruit him here, and this first year has been really, really wonderful. Before arriving here, he was associate professor of pediatrics at Vanderbilt University, and then appointed as chief of both UCSD and Rady's Children's Hospital neonatology divisions in 2013, and did a complete merger of a previously separate group, and really extraordinary contributions to that program. He is a multidisciplinary and collaborative investigator that enables him to address one of the most intractable challenges in neonatology, which is BPD, or bronchopulmonary dysplasia, in babies born extremely preterm. And his NIH-funded laboratory is focused on molecular and cellular mechanisms controlling lung development, and I'm sure we'll hear much about this. So he has a long and distinguished career mentoring clinical and scientific students and trainees, many who have gone on to establish their own successful research careers. He's really a dedicated, tireless, passionate, supportive, and generous lifelong mentor whose joy for discovery serves to inspire scientists and pediatricians at all levels. And we're so pleased that we have an opportunity to hear that. So thank you, Lance.
Thank you, Mary. That was very kind. So I think I've probably given away my bias in the kind of the punchline of my talk, as far as my particular interest in understanding molecular mechanisms of disease, and I'll tell you right up front that I'm going to try to make the case that we need to leverage our new advances in technology and in science to try to find a better way to take care of babies in the NICU. So, but first, I think it's worth actually taking a little bit of a survey of what type of babies end up in the NICU. So if we're going to make a case for using new technologies and experimental therapies and all this stuff, we really need to know what we're talking about. So there are a number of reasons why babies are end up in the NICU. Some of them will have structural birth defects, and a couple examples here are a drawing of a hypoplastic left heart. A lot of these babies are first diagnosed or stabilized in the neonatal ICU before going over to the cardiovascular ICU or having surgery. And most of you will probably recognize that the chest X-ray on the right is not normal. The right or the left hemithorax is full of bowel loops, and this is a baby who has congenital diaphragmatic hernia, and those continue to be some of the sickest infants that we care for. So there's structural birth defects is one class. Also, there are a lot of babies that are affected by injury or infection. So the baby on the left has had profound hypoxic ischemic encephalopathy, which is typically caused by lack of blood flow and or oxygen delivery to the brain around the time of birth. The baby on the right is being treated for septic shock from gram-negative bacteremia. So these are certainly types of cases that we have in our unit. We also have a myriad of collection of various metabolic diseases, if you will, everything from congenital disorders of glycosylation, it's just kind of schematized on the left, to disorders of ion channel function and cell physiology, in this case on the right, a diagram of what causes long QT syndrome. But the vast majority of babies end up in our NICUs, and this is around the world, are those that are born preterm. And so most of you may know these numbers, but I'm still a little bit amazed at the scope of this problem. So 15 million babies are born preterm each year, and this is from the latest WHO survey that I could find. And we're talking about 1 million deaths a year around the world. 1 million babies die a year from preterm birth. And not only is it the leading cause of death in newborns, it's actually the leading cause of death in children all the way up to the age of 5. So the impact of this is huge. I don't think I need to sell anybody on that. So the question is, how are we doing with this? And so, is this isolated, or is this a problem that the United States has, or whatever? So this just gives you the preterm births in each of the top countries around the world. And you can see the scope of this. And, it's not like the US is doing as well as we should, given our technology and our wealth. So how are we doing though? I mean, we've had NICUs in the United States now for over 50 years. It's been a growing field, a lot of new technology, a lot of advances, a lot of attention given to the problem, and a lot of people are working very hard to try to improve outcomes. And so how are we doing with all that progress? So these are data from the Vermont Oxford Network, which is a now international group of NICUs and grouped by acuity and class, if you will. And those data are compiled on a regular basis each year. And this is actually showing one of the key performance measures that are actually included in this. And so this is mortality of infants born less than 1,500 grams. And it's broken down by quartiles. So you can see in the light green, this is the median, 50th percentile. This is, right in the middle of how we're doing around the Vermont Oxford Network. But I also like to include, these are the best 25%, the best quartile. And this is the 75th percentile. So the different quartiles, so you can see what the range is. So over the last 15 years, the mortality in kids born less than 1,500 grams, if you look in the middle, has been between 10 and 12%. And there was a little bit of improvement here for a while, and that would have been great if that would have continued. You can extrapolate down to maybe 5%, but it stabilized and actually has ticked back up a little bit in the last few years. I don't know why, I don't know what happened in the last couple of years, but these are the data. And these are, again, from state-of-the-art academic, cutting-edge places, including with all the other types of really, high-level NICUs around the area. This is severe intraventricular hemorrhage, and we know that this is one of the major risk factors for poor neurodevelopmental outcome. And again, you can see if you look at the middle quartile, the 50th percentile, we were doing a little bit better between 2005 and 2010, went from 10% down to 8%. And since then, it's somewhat stabilized. A little bit better, maybe. Retinopathy of prematurity, obviously the major cause of blindness in babies that end up in the NICU for prematurity. Again, doing maybe a little bit better, but somewhat stabilized over this period of time. And this is bronchopulmonary dysplasia. So here I am, the guy who's been studying this in the lab for more decades than I care to admit, and actually I have a very big interest in improving the clinical care of BPD as well. And have we done anything with the incidence of it? Well, not really. You know, if you look at the last 15 years, it's pretty stable. And, you know, this is a disease that was first characterized here at Stanford. I mean, it's something we should have a lot of pride in actually trying to eradicate from the planet. But for whatever reason, we haven't really made a lot of progress in the last 15 years. And this is with new ventilators, CPAP, new surfactants, new data around surfactant, all sorts of bundles that we've tried to put in place to try to improve outcomes. The reality is it's still a major problem. And it's not just the Vermont Oxford network. So these are you know, different international networks for neonatology. These just show the outcomes back to 2007. It's a composite outcome with and without bronchopulmonary dysplasia, just to kind of pull that out, since that's one of the more common ones. And you can see that each country is seeing something kind of similar, maybe some improvements here and there, maybe a few places where the outcomes are getting a tad worse, but for the most part, pretty flat. And this is, and I say flat, we're talking about a situation that causes 1 million deaths a year around the world. It's the #1 cause of death in kids less than 5. And over the last 15 years, we really haven't made a lot of progress. Maybe a little, maybe we're getting better. And I've seen some data suggesting that overall neurodevelopmental outcomes in our survivors are probably a little better. So that's very reassuring. Hopefully, you know, we're doing the right thing there. But I think there's an opportunity here for us to do a lot better. And that's what I'm going to try to convince you of today, is that I think if we are thoughtful and if we are aggressive and open-minded, that the future could be a lot better and we could make some real inroads into how we care for these kids. So we've been always good in neonatology about getting information. So this young attending neonatologist here is examining a patient, but also, as we tend to do in the NICU, looking at the monitors. And you can tell we've always been pretty good in the NICU about putting lots of machinery and monitors in there. And it's a really amazing what we can capture from a small infant regarding their physiology, real time. We can look at organ dysfunction, we can look at response to intervention, and we've been developing these technologies for, again, many decades, and we're getting better and better at them. And so there's an opportunity there. However, I think we need to start changing how we use them. And instead of just actually measuring physiology on a real-time basis and deciding how much blood pressure medication or how much oxygen we need to give a baby using this information, we need to start figuring out a way how we can take all this information and actually see how well human development is going. You know, how are our babies doing as far as real growth and development? How can we measure repair following injury, which unfortunately a lot of our babies suffer injury from just exposure to the environment or infection, sometimes real injury, and we'll get into that a little bit. But how do we know how they're doing following that? How do we use this information to project disease risk? And if, let's say we find a new treatment or a new cure for something, how are we going to know that it's working? And can we use this non-invasive tradition of monitoring and information that we've developed in neonatology really to see and understand how our efforts are actually progressing? So some of this work's been pioneered here. And so down on the right, you see Susan Hintz, Vinny Bhutani, and Valerie Chalk. Among all the many things that group has done, I wanted to highlight a little bit near-infrared spectroscopy. This is not brand new, but we're starting to learn more and more about how it impacts clinical care, and it tells us a little bit more about what's going on with our babies. So it's basically using near-infrared light to measure oxygen saturation deep within the tissue. And deep enough that we can actually measure oxygen saturation inside the brain, inside the cortex, inside the splanchnic bed. We can get an idea of what's going on in the kidneys. And this is actually now, here, not everywhere, but here, it's actually part of routine clinical care, and both in the NICU and in other units as well. And I think it's really, really valuable and really important. And I think it's just kind of scratching the tip of the iceberg, if you will, as far as what this could be used for and what these types of approaches could tell us. So Valerie is not alone in the division. So there's a picture of her and Krista Van Meurs and Shazia Bombal and Sonia Bonifacio, Melissa Scala. They're all interested in taking the different modes that we have, new technologies, and really trying to understand what makes our babies sick, what can make them better in a way that maybe people haven't done before. And this, it spans everything from hypoxic ischemic encephalopathy, its treatment, how you transport kids when they're really sick to a place like Stanford, how do you actually image what's happened to their brain, how well it's developing, and how are they developing overall when exposed to the various things that unfortunately our babies are exposed to in the NICU, like painful procedures, alarms, poor sleep, not being held by their mom enough, all these types of things. So they're important in their overall well-being. There's some new technology coming down. So one of my good friends and now a colleague here at Stanford, Todd Coleman, has developed a non-invasive way to measure motility in electrical waves through the stomach. And starting out mainly with, think about the situation of diabetic gastroparesis in adults. The problem with measuring, gastrointestinal waves in a non-invasive way is all the noise. There's so many different electrical signals that can be measured on the abdomen, what really helped Todd's crew do this was a higher order analytic approach to filtering the different signals that they were perceiving, and they were actually able to then get through and just detect the electrical signals coming through the stomach and how the stomach empties. And you can see how it works in adults. They have made a lot of inroads. Now they have coupled this type of an approach with the development of flexible temporary tattoo sensors, if you will, that his lab has done a lot of great work on. And I know he's got a prototype that works on babies. And so the question is, can we use this type of an approach, not only to look at, say, the stomach and the waves of peristalsis and electrical signals in the stomach, but the rest of the GI tract, knowing that each part of the GI tract has kind of its own pacemaker signal. Can we learn about motility in preterm infants or in term infants? And when babies are in the NICU, how that is normal or abnormal and how it develops with time. And more importantly, can we help it? Can we improve it using approaches like this to actually know what we're doing? So there's a lot of new technology out there as far as monitoring. And my hope is that not only will it allow us to measure vital signs better, but it will allow us to measure the things that we think we need to know to see if we can actually do a better job in taking care of some of these kids. And let's be honest, it's got to be scalable, right? 15 million preterm births worldwide. It doesn't do us any good to have something that only works in the Stanford NICU, right? So it's got to be something that's scalable, it can be widespread, and can have a large impact. So now I'm going to go from something kind of large and physiological to something very small and very complex, and that's the genome. And I'm going to share my bias again here, that I'm a big fan of genomic sequencing of babies in the NICU. Everyone in the NICU knows this. They've heard me kind of go off on my tangents about them quite a bit. But I think that there's a lot of value here. And I'll say right up front, I believe that every human should own the information of their genome. They should have control over it. And the babies in the NICU and their families should have control over that genomic information. I think they should have it. And hopefully they should share it with their caregivers when we need it. And I think that there's a lot of research opportunities in here too. So I'm hoping that we can, over time, navigate some of these concerns, potential concerns, and just make sure that genomic information is treated like any other biologic or clinical piece of information that helps us take better care of our patients. So, I've been exposed to this in the team down at San Diego. We've been sequencing genomes in the NICU there many years. I know they've been doing it here too as well. And so one of the big advances that happened down in the crew in San Diego through a collaboration between Rady Children's and Illumina was just kind of making the whole process go faster. And so there are two things I want to point out here of these two workflows. The first one, actually at the top, we get a sequence of genome pretty quickly, but the one on the bottom got it sequenced in a day. And so one of the big differences was going from the HiSeq 2500 to Illumina's new NovaSeq 6000, just allowed you to have more capacity, and we could sequence more genomes more quickly. And there were a whole bunch of those humming all the time in our Genomic Institute. But one of the other parts was going to the EHR versus using a different approach. And so when we first started sequencing genomes in San Diego, it was clear to all of us that we needed to do a better job phenotyping our patients. Hypoglycemia alone would not tell us exactly what was going on and what variants we needed to be looking for. Same with hypoxia or whatever it is. And so one of the ideas I had was, let's go back to Epic and let's make it much more, database driven in how we put in information, using NoteWriter more, using more discrete data fields. And as most of you know that have tried to do things like this in Epic, it's done. It can be done. It's feasible. If it's a little slow and a little cumbersome, and in the end, it really wasn't making much progress. And so the informatics team actually developed a natural language processing approach where they basically said, you guys can type in whatever you want. Don't change your notes at all. Do your problem list, do your diagnoses. The computers will mine it. And so they were basically mining the EHR for information that might help connect variant calls to actual clinical scenarios. And that was a huge improvement in the rate at which we could actually get meaningful information from genomes. This is kind of what a schematic looks like. And so basically, so for example, this patient was admitted for poor feeding, ketonuria, and metabolic acidosis. The natural language processing can go through all the different parts of the EHR, get all the different aspects of that might complement it or might clarify, and then be able to take all the variants that were called at the end of sequencing and then match them up. And I think this was actually a case of maple syrup urine disease that was picked up by sequencing within a day. And so it works. This is Sebastiana. She was our second kid that we sequenced with intractable seizures. The first one had been having seizures for probably a week, wasn't responding very well to therapy. Then I finally got her genome sequenced, and it took a couple of days to get the results. I think it took us 4 days to get that call. The process continued to improve. We had Sebastiana's genome done at day 2 of her life. And she had an ion channel mutation, was switched over to Tegretol and Keppra, and her seizures were controlled, and they still stay controlled to this day. She would come around the Genomics Institute offices every once in a while and just say hi to everybody with her family, and she's doing very well. So we had a kid with quote unquote intractable seizures who was still seizing on 3 medications, the 3 we usually give in the NICU. And by day 2, though, by day 2 of life, we had her diagnosis and we had her on the right therapy. And that's just one case. So this was something that was happening almost on a daily basis. It also helps in things that I honestly didn't think it would. And so this just highlights a group of patients with congenital heart disease in our NICU. And this study was actually led by Natalie Sweeney, a Stanford alum. And what she found is about half the kids with congenital heart disease in this cohort, there was a variant that was identified. Most of the cases, there are the sets of trials we've done, it's about a third, but in congenital heart disease cases, it was half. And sometimes it helped us just understand the diagnosis. There were a few cases that by getting the genomic diagnosis, it led us to recommend palliative care and be able to do it much more quickly than maybe we would have in the past. There were a couple of interesting stories, though. Like there was one kid who was having calcium and phosphorus abnormalities and was diagnosed with X-linked hypophosphatemic syndrome and was put on the right therapy and was stabilized there. And so before bony changes were detected. So I think that's pretty good. There was also a kid who had congenital heart disease. I think it had been repaired, or at least a palliation, if I remember right, and still had severe cholestasis. And they actually did a liver biopsy, and it showed, a really abnormal finding. Could be consistent with a biliary atresia. and just with hyperproliferation, all the small bowel ducts. And so the kid was actually scheduled for a Kasai procedure, and it was the morning of surgery. I kid you not, that morning we got the diagnosis back of a JAG1 mutation. So the kid had Alagille syndrome and didn't have all the other classic signs, because Alagille has variable penetrance. But the reassuring thing there is that the cholestasis and liver problems and Alagilles at birth often resolved on their own. And so this kid avoided a Kasai procedure, like with hours to spare, literally. And slowly got better just with conservative management. So it works. So if you get genomic diagnoses on kids, you can improve their care. Our experience has been about a third of the time we get a diagnosis. It's a diagnosis with a documented in the literature variant that correlates with their presenting signs. And about half of those kids, we actually change the way we manage them. So if we're going to do this on all kids, we need to be thoughtful about how much it costs, whether or not it's scalable, like I said. And so one of the studies that the team down at Rady did was this Project Baby Bear, where they did 5 different children's hospitals around the state, looking at getting the state to fund it, just looking at the overall impact of it, and could it be replicated? And again, still about a third of the time we got an answer. And then the cost savings per child was interesting. What they actually did this, they looked at rapid whole genome sequencing versus maybe the typical turnaround costs. And what I wanted to highlight here is the cost savings per child. And there's a low and a high range here, because depending on how much the impact of the sequence and this answer changes their clinical care. So obviously there's going to be a range. But per child, getting a rapid whole genome sequence 3-day turnaround saved between $12,000 to $15,000 per case. The longer you go out, and so if you send it through maybe routine sequencing and analysis pipelines, 7 days or 14 days, the cost savings is much less. And at the time this study was done, it was costing us about $8,500, I think, maybe $9,000 to do a genome. So it saves money. And that's why insurance companies are approving it. That's why the state is hopefully going to start approving it. It's why we need to be doing it more. I want to go ahead and be very open though about the inclusion and exclusion criteria here. And so these were the inclusion criteria for this study. And I want to highlight a couple things on the exclusion. So we have not been routinely sequencing the genomes for kids that are in the NICU only for prematurity and without any other complications or other signs that don't seem to completely normal. Infection and sepsis with normal response to therapy, that was excluded. HIE with a clear precipitating event. Now that gets a little bit tricky, because that clear precipitating event is sometimes hard. And they're actually doing a study now where they're taking all kids with HIE on a national study to look at their genomic sequencing to see if that helps at all. And a few other things that we think are probably, probably not related to genomics. My argument is that until we sequence the genome of all of these kids too, and see what kind of variants they have and how that might impact their clinical course, we don't know if there's going to be high yield there or not. So I think that that's going to be the next challenge, to really see where does the benefit extend. We have a great team here at Stanford that is doing these studies all the time too, and the group down in San Diego benefited a lot from the collaborations up here, and so I think that we have done some great work. I'm excited to see that John's going to be presenting on the Undiagnosed Disease Network study coming up soon. I look forward to that. But I think that we actually have the potential here to do rapid genome sequencing on critically ill patients, not just in the NICU, but around the hospital in a faster and more clinically impactful way than anyone has done it before. And so we are poised and well positioned to be able to bring this technology directly into the bedside and have it make a big difference in how we care for patients. So one of the things I haven't yet talked about is this idea of, what do you sequence? And so most of our understanding about, how genetic variants affect, physiology or biological function, has been based on the central dogma of biology, right? That you get a change in the DNA, that leads to a change in the mRNA sequence, and you get a peptide mutation. And that mutation in that peptide, that protein, affects how well enzymes work, or whether or not receptors can actually bind to their ligand and signal appropriately, whether or not a transcription factor can function. All of these things leading to disease, right? Well, it's obviously not that simple, because all of this quote unquote dark DNA and junk DNA is rich with important features. And so one of the things that personally I've been more and more interested of late in the lab, and I feel embarrassed that we weren't studying this 20 years ago, are gene enhancer regions. And so these are regions of the DNA that do not code for protein, not made into RNA. They could be millions of bases away, right? But they contain regions that bind transcription factors, and in binding these transcription factors controls the gene transcription. And there are a lot of variants in these regions. And so here's an example of a variant that can cause relative changes in how well this enhancer works. And so it can change the transcription, and you get difference in message levels, even though it's not a coding variant. You can also change whether or not that area of the DNA is has open chromatin or is silenced by epigenetic changes. And so it affects gene regulation in that way too. These are very common throughout our genomes. And there are many, many biological examples, including examples where changes in an enhancer can affect how well a gene is expressed in different cell types. So here's 3 different generic cells. The same enhancer region will bind different transcription factors in those different cells, leading to give different levels of gene expression and a different impact of that variant. And we see this in the NICU all the time. I don't know if we always appreciate it, but there are so many situations where we probably or definitely have a genetic variant that affects every organ differently. And one of the things I love to kind of think about, because it's just hard to comprehend, are ciliary diseases. So remember, every cell in the body has a primary cilia, or at least does at one point in its life. And that's addition to the motile cilia that you think about in ciliated cells. And these cilia are important for lots of signaling pathways. The number of signaling pathways that use cilia or localize their receptors or components to the cilia is really unbelievable. Anything Wnt signaling, TGF-beta signaling, mTOR signaling, NF-kappa B, of course, sonic hedgehog signaling is very, very tightly regulated by ciliary function. And there's more. And look at all the different syndromes that we see that have cilia at their kind of basis. And every one of these kids has a little bit different manifestation of the disease in the different organs. And so not only do we really need to know the whole genome in our patients, we need to start thinking about how do we understand the somatic impact of those variants. What is it doing in the heart versus the brain versus the kidney? And are there ways that we can measure that? Or based on the actual, can we just actually take the variant calls off of the DNA sequence from any tissue or any sample, and then know enough to understand how it might be impacted in different tissues? It could be really, really important. So it's not just the genome though. So one of the things that I'm constantly trying to remind trainees is that most of our kids are not diseased at the day they enter the NICU. It's that they're immature. And during the course of normal human development, they go off track. And their development doesn't progress as normally as it might if they'd stayed healthy inside mom. And that's when we get a lot of the complications of prematurity. So this gets really complicated, right? So everyone has their genome, which kind of lays the blueprint of the different variants they have, which allows them to have all the features of who they are and how they might respond and be healthy or not. But then there's all the things that are related to that. There's, of course, measuring proteins production, which is important in the proteome, environmental influences, which are huge in the NICU, because obviously the environment in the NICU, although the best we try, is never going to be like it is in mom. The metabolome, which is a much larger idea than what the name gives it credit for, because thinking about it's not just metabolism in a simplistic way about how well mitochondria work, whether or not you're using glycolysis or not. It's all the different molecules that are produced within the body that regulate physiology, function, and development. And so you can imagine how complex that gets. You can focus just on lipids, you can focus just on steroids, amino acids, energy metabolites, mitochondrial metabolites, you can imagine. It's very, very complicated. All of these contribute to the patient's phenotype. So luckily, we can measure all of these things now. And so this is a slide that was shared to me from the crew here. The team here at Stanford routinely measures all of these different things. Not only the sequencing the genome, measuring immune cell function and identity, looking at proteomes and the metabolome, looking at the microbiome. Not only the microbiome as far as which bacteria are in your gut or on your skin or in any other organ, but what are they doing? What are they making? How are they influencing your own body? The epigenome, which is, I hinted at a little bit, whether or not you have open chromatin or closed chromatin throughout your body, whether or not that's accessible, or what the methylation and acetylation patterns are, whether or not that just goes with aging, or whether or not it's something that's plastic. And of course, the transcriptome, which is even harder to measure clinically, except for maybe blood samples, and actually seeing which mRNA molecules are truly made. And then, like I said, the expososome, which for us might be pollution and the things we encounter. Kids in the NICU, it's obviously something very, very different. So this just kind of highlights some of the people here that are doing these kind of work. So we're getting this information now, and we are starting to learn more about how all of these data interact and both in different patients and how they can kind of correlate with clinical outcomes. And it takes a huge team to do this type of science. It takes a lot of really great science, a lot of integration, a lot of informatics. And so these are huge data sets, and it's how we put them together and match them up with clinical scenarios that I think is really important. Just want to give a few examples here. So this Carl Sylvester's team has done some really nice work, mainly focused around necrotizing enterocolitis in the NICU. And this is just showing a sample of data, some things that they found in longitudinal studies of patients in the NICU that either end up getting NEC or not, and trying to identify some molecular signatures of this disease risk. Nima Agipur and his group have kind of challenged what we know about, when does a baby term or when are they preterm, and looking at the complications of prematurity. And I think we probably all knew this or sensed it, but Nima's group did a really nice job at actually measuring it. And so instead of trying to draw these different boxes and lines, which is say 28 weeks, 37 weeks, here's the actual distribution of patients, and their complications. And you can see, yeah, there's a lot more, kids that are, older who are healthy and not in any complications. And then some of these complications of prematurity are a little bit more prevalent, lower. But obviously there's gradients, and every patient is unique. And we always see a patient that's a little older than, and it gets a complication of prematurity, and we think that's kind of weird. We didn't expect that. And then we see some kids that are very immature that do quite well, and they're very resilient to disease and don't have those complications. Here's an example. I love kind of walking through some of these models. And so, for example, they took this whole large data set of information of all the metabolomic information they got over time, and then looked at some of the different components of that, tyrosine metabolism, different fatty acids, free carnitine. And by building a model, they were actually able then to develop these subgroups of risk of different complications. And this is another example of some of the more common pathways that were more likely to be seen in patients developing these different disease risks, being born premature. The amazing thing is that they can get some of this information from mom before she delivers. They can get some of this information at birth, and some of this information after birth. So, and after, as a kid is in the NICU. And so there really is a continuum here of potential risk and changes in metabolism, and setting up patients for disease processes that we really don't yet understand. But now we're starting to kind of, again, scratch the surface a little bit. And so the next thing would be to try to see is, well, how can we help this? How can we intervene? And this is actually, an illustration of another project where, looking at everything within the EHR and all the information we get, all the labs, all the procedures of moms while they're pregnant up until birth, and then afterwards, and then kind of putting this all together and finding potential exposures or treatments that mom receives before she delivers around that time, and really trying to understand things in a way that maybe we didn't in the past. I think, and that's the important thing. I think we need to own the fact that we can do better and have an open mind about How are we managing our patients? And is there a way we can do better? Here's another wide open field, I think, and this is the gut-brain axis. This is the kind of diagram. So again, we know that the microbiome is important. And so the more and more we learn about the microbiome, the more we think that it's probably controlling everything. But one of the things that the gut microbiome does is it actually does play a role in production of neurotransmitters in the body. And so those neurotransmitters from the gut do actually go to the brain. And of course, the brain then has influence back on the gut as well. And so the question is, how does this happen during development? You know, we've mainly been studied in adults and older children, but what's going on in a preterm infant? How is this system working? And is there a way to make sure that it happens the right way? Or if it's not happening the right way, can we correct it? This paper just came out in Cell, and I just showed the kind of the diagram from it. This is a really intriguing idea that the immune system has memory that's hardwired in the cortex. So in this study, what they showed is that severe inflammatory processes in the peritoneum or in the gut, actually the response to that inflammation was stored within neurons in the insular cortex. And then after the inflammation had resolved, those neurons, when activated, could replicate peripheral inflammation. So those mice actually got peritonitis or colitis without any injury to their peripheral body, just by activating that part of the brain. So it is all in your head, and it's also everywhere else. And so finding out these mechanisms, I think, is really important. And again, how does this happen when your cortex is still developing? Babies in the NICU often are exposed to multiple inflammatory episodes. Well, maybe, are they really all infectious, or are some of these actually immune memory that's coming back? I don't know. So we owe it to them to try to figure this out. So again, it's about understanding how we can avoid these complications of prematurity by ensuring that we get normal development along this lifespan. And so in my lab, we're interested in lung development. And so, you know, the lung is very immature in a lot of the kids that are admitted to the NICU for prematurity. And in a lot of these kids, the overall lung maturation fails to occur normally, and they end up with bronchopulmonary dysplasia. Well, we've learned over the years that inflammation is a major player in this disease. And what it probably does, and rather than injuring the lung, it actually just basically kind of removes the normal developmental wiring system for lung development. And so when the lung responds to inflammation, the signaling pathways now are not conducive for normal development. And macrophages are the key inflammatory cell that does this. And the macrophages in the lung of a preterm infant are not the same as the macrophages in the lung of an adult. And so we did a large study looking at babies in the NICU who are intubated, looking at their lung macrophage gene expression, and just kind of going through the data, we found a couple of interesting patterns. And I'm just showing one gene. This is macrophage scavenger receptor 1. And what we found is that, you know, we had a fair number of kids who were very immature on the ventilator for several weeks, who ended up not getting BPD, or had very mild disease. So they were off oxygen by the time they went home. And the expression of this gene MSR1 is interesting because it's pretty low at day 1, but then it goes up by day 7 and it stays up kind of. But in the kids with severe bronchopulmonary dysplasia, the pattern was different. It didn't go up as much between day 1 and day 7. It was more scatter, and it stayed kind of low. And it wasn't alone. There were other genes with this pattern as well. And without knowing any better, we called these resilience genes. Genes that correlate with a baby's ability to avoid bronchopulmonary dysplasia, that go up between day 1 and day 7. So I was actually dumb enough to start going through all of our data, looking for this pattern one at a time. Luckily, Debashis Sahu down in San Diego, who another Stanford alum, had a better idea. And he uses Boolean approaches to actually mine large data sets. And he said we can use a program that he developed called StepMiner to look for these different patterns, to see when genes go up or down over time and see if that correlates with outcome. And that's what he found. And so this is an example of these resilient genes. So these are the kids who got either no BPD or a mild disease. A lot of genes went up between day 1 and day 7, like I showed you. There was also a few genes that went up between 2 and 3 weeks of life, and then another set of genes that went down after the first day of life. And so you could look at these, and now we're trying to mine this data set to look at what those genes are. When we looked at the kids with severe bronchopulmonary dysplasia, there were fewer genes, and the patterns weren't quite as clear, but they were still all there. Okay, so maybe they're just doing the same thing, just to a different degree. But when we looked at the genes, the actual genes, they're very different. Of all the genes that have these patterns, only 58 were shared between kids with resilient to disease and kids with severe bronchopulmonary dysplasia. And if we take the genes that had a pattern over time in the kids who didn't develop severe BPD, and we looked at their expression in the severe BPD patients, this is what you see. It's pretty much low throughout time. So those kids don't acquire the expression of those quote unquote resilience genes. Well, so what? Well, we are interested in how genes become transcribed or not. And so again, it goes back to enhancer regions, promoter regions, transcription factors that mediate the expression of all these different genes. And if we could identify how this works, how these genes are turned on, maybe then there's a therapeutic option. And so one of the first things we did is we looked at the enhancer regions and promoter regions that were predicted to bind transcription factors in these different gene sets. And this, we saw some differences. And so the kids who are quote unquote resilient, the genes that they expressed uniquely over time are predicted to bind this set of transcription factors in their promoter enhancer regions. And these are not necessarily transcription factors that are known to regulate gene expression in macrophages. There's a few interesting stories in here. NFAT-C2 is really cool because its role in regulating morphogenesis of a lot of different organs and health there. But we're actually going back and doing the biology to try to understand how do you activate these transcription factors? What sort of pathways are involved? In the kids with severe BPD, NF-kappa B activation we knew was important for inflammation. And we had some interesting data in mice that the connections between NF-kappa B and SP proteins might be playing a role in hijacking normal developmental mechanisms. So that's an interesting thing that we're looking at as well. And then the role of MYC and MYC signaling and MYC-mediated transcription is also something that really hasn't been studied in BPD pathogenesis in detail, but we're going to look at that now. So the idea is to take all this information, all these different multi-omic studies, and bring it back to something that tastes as good as amoxicillin, and we can just give it to every single kid that needs it. And then hopefully by thinking about this and trying to take a new approach, we can do better. And so I think that that's the challenge, and that we all as a field own it, and that we have to try to think about how we're doing things and what it is that we don't know, and how can we challenge ourselves to do better for our kids. So thank you very much, and happy to answer any questions.
Thanks, Lance. Sorry, it was a little technical difficulty. Okay, well, first of all, I thought that was extraordinary for many reasons. So the first is that I'm track on the participants, and you've reached participants all over the department and in the school, far and wide. I also knew how amazing your work was as relates to lung disease. And I knew there was so much enthusiasm, Mark Nichols and Tushar Desai and others in getting you here, but the reach has been incredible. I'm also so happy to see how it connects to the UDN. I think the fact that you said a third of patients had a genetic diagnosis, it was really informative. And I think that's about the rate that the UDN sees, with half going on to have changes in, therapy because of it. The other thing is, one of the key elements of the integrated strategic plan across all of Stanford medicine is uniquely Stanford, and the number of investigators that your work touches, I think, is a great example of that. So next year when the dean asks me for examples, I know which one will be front and center. Okay, so we had some good questions. The first is, could the EEG technique be used to monitor and prevent NEC? I think that question came early, and you did talk about NEC a little bit later, but maybe expand on that.
That's the ultimate goal. I think initially we were just going to see if could it connect with feeding tolerance, right? So whether or not babies have normal motility, could we then advance feeds more quickly in some kids versus others? And then whether or not motility of the GI tract and coordinated motility plays a role in the pathogenesis of NEC, we don't know yet. So that's exactly the sort of thing that we need to figure out. And it may or it may not, but regardless, it's something we have the potential now to actually test and actually see what we can learn.
Okay, great. Why only Medi-Cal patients in Project Baby Bear?
It was funded by the state for that reason. So
Oh,
that's just,
great.
that particular project was a state-funded study. But there have been a lot of other studies that have found pretty similar or comparable results. Remembering that the cost of sequencing comes down every day, and so that part of it is always going to move. But it's not just the sequencing, right? It's also the cost is also the interpretation, the variant calls, making sure that you have the infrastructure to have this be actionable.
Oh, great. Okay. Anyone at Stanford looking at heterogeneity in intracellular motors and kinesins as it affects phenotypes?
Ooh, I don't know if anybody knows of anybody. Let me know. I think
Yeah.
that would I think that would be really, really cool. I mean, and just again, I remember when one of my colleagues who was studying, he was actually studying polycystic kidney disease, and he was trying to convince me that cilia were important in every single cell in the body. And I was like, yeah, whatever. Cilia just clear mucus. That's all they do. But he kept showing me this data, and then the field started to really grow. And I was like, well, I feel pretty stupid. You're right. It's biologically, it's really interesting to think about how all signaling doesn't just happen globally within a cell. It's always localized to these specific areas. And if you think about cilia are important for determining the very, very first steps of sightedness in the development of the embryo, then all the other signaling pathways that they play a role in. It's really, really interesting. I'd love to hear more about what people are doing here in that area.
Yeah, so that comment came from an anonymous attendee, so maybe they'll show themselves. The other thing is you mentioned how important cilia are to kidneys. They're actually incredibly important in the bone, where they are the mechanosensors that sense weight-bearing activity. So that's great. Has anyone looked at the genes that were identified in the severe BPD group to see if these are present in patients regardless of age with severe COVID-19, regardless of perinatal history?
I don't know. That's interesting. A lot of the genes that we see that are higher in the severe BPD patients, a lot of them are inflammatory genes, right? So they're inflammatory mediators. However, it's never quite that simple, right? It's not just inflammatory mediators, because you think about the case, we were very interested in IL-1 beta and continue to be. IL-1 beta has some therapeutic, drugs that you can intervene with, but IL-1 beta is also one of the major signals of telling the immune system to shut off and promote eventual repair. So if you completely block IL-1 beta, you end up running the risk of having persistent inflammation, even though you may not get such a severe initial one. And then one of the things that really fascinates me is it seems like every microbe has their own different inflammatory signature that it can potentially cause. And it's not the microbe, it's the host generally, right? It's our inability to kill off whatever microbe that we don't want and then resolve inflammation normally. And I think, that's why all the initial attempts to lump COVID in with quote unquote cytokine storm, like you would see in HLH patients, didn't fit. It was a little different. And it's the dysregulation of, say, interferon versus some other pathways that are giving it its unique disease, and the cellular side too, right? It tends to affect the vasculature more. So I didn't answer your question. We don't yet know if there is any overlap between BPD pathogenesis and COVID syndrome and the inflammatory syndrome in the lungs after COVID. There's going to be some overlap because there's such immense inflammation. But as far as anything more specific than that, I don't know. But it's a really interesting idea.
Okay, and revealing that I'm a card-carrying bonehead, IL-1 beta is important in bone.
Yeah.
Okay, all right, good. Oops, I think we have one more question. All right, I think these are amazing questions, Lance, in terms of how transdisciplinary they are. Are there any peculiarities about mitochondrial genomics that make it more or less useful for this type of scrutiny and potential interventions compared to nuclear genomics?
Yeah, wow. I don't know. It's really intriguing. I mean, there are ways that you can look at the mitochondrial genome now, as most people know, and the inheritance in the mitochondrial genome is different, right? So that opens up some interesting paradigms, and it's got to, we know it. There are kids that have mitochondrial disorders, right? So that's all known. The impact of that, though, and how we do it routinely, I don't know. I think that's a really interesting idea, and it also kind of fits with the part of the metabolism that we generally don't get into. And that is, the cells in the body that are, using their mitochondria and mitophagy and ones that aren't. And then in the area that I work in, a lot of these inflammatory macrophages practice aerobic glycolysis, the Wardenberg effect, and they don't use their mitochondria when they're activated because they want to, quote unquote, preserve biomolecules for inflammation. I don't know what that means, but there's a lot that needs to be understood there. But that's intriguing, because it would be really interesting to look at mitochondrial genomics in kids who don't have obvious mitochondrial disorders, because it would be really interesting to learn about what might be there.
Great. I think that's amazing. And I can't think of a grand rounds that sparked so many new collaborations. So I hope everybody that's interested in this will be quick to contact you. I think your dance card's going to be very full.
Great. Come on.
All right.
I'll show you around.
Yeah. Good. All right. That's the end of our hour. Thank you so much, Lance. Really a pleasure.
Thank you.
Thank you, everybody. Have a good day.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.